Thank you. Thank you very much. I appreciate it. And I want to thank for the invitation. It's a pleasure to be in California after a short trip of about 20 hours from Jerusalem. And I'll try in my talk, if I know how to advance my slides. Anybody can show me how to advance my slides?
We have a little pointer up here. That's right here should, now, I think that you can point there and it will give you a laser pointer which will show up behind you. And I think
how do you advance them?
maybe the top button.
Yeah.
There we go. Top for forward,
okay.
bottom for reverse.
Okay. I'll try to present an overview of things that are happening in the cannabinoid field. For me, for my group, it has been quite a few years. As a matter of fact, as one mentioned before, we started work in the early 1960s. I believe that many of you were not born there. And thanks. When we started work, there was absolutely no interest. As a matter of fact, when I asked for a grant from NIH, and NIH had a lot of money at that time and gave a lot of grants, they told me, and I knew the people there, there were many of them outstanding scientists, when you have something more relevant to the US, contact us. Nobody smokes marijuana. It's something that's being used in Mexico. Well, a year later, or 2 years later, their pharmacologist came back, and I asked him what happened. Somebody important, maybe a senator, had approached them and had asked whether marijuana destroys the mind, the brain. Apparently his son had been seen smoking pot, and he wanted to know what's going on. Well, things have changed since then. Nature, probably one of the most important scientific journals, Nature published last year 4 reviews, and this is our review here. You can see the brain and the cannabis, and all the time we have additional information. And the reason for that is, in the medical side at least, that it is known today that modulating endocannabinoid activity may have therapeutic potential in almost all diseases affecting humans. And this was published, this was in a review by 2 scientists at NIH. And while I'm not sure that this is absolutely true, affecting all diseases affecting humans, there is no doubt that quite a lot of diseases are affected, and therefore there is a lot of interest, contrary to what happened, as I told you, a few years before. But let's go back and look at some of the interest in cannabis. In my area of the world, in the Middle East, it was used for thousands of years, and this is something that the Assyrians had. And they had 3 types, so they use it in 3 ways. They used it as a recreational drug, if you wish, in modern terms, and they call that ganzigunu. They use it apparently for neurological diseases, and they call that azalo, and they apparently it was also part of their religion. And this went on for quite a lot of years, and this is something from Dr. Reynolds, who was the personal physician of Queen Victoria. Queen Victoria had severe migraines, and he used to bring cannabis from India. Apparently, European cannabis was not good enough. He used to bring cannabis from India for her migraines, and he didn't mention her name in her articles, but it was well known that this is what he was actually involved. When we speak today of cannabis, most people will think of the plant alone and the compounds that are found in the plant. And I'll, of course, speak about those. But it is not so simple, and things have developed quite rapidly over the last 20 years. And I can see 3 phases of cannabinoid research. One of them has to do with the plant, the phytocannabinoid research. The second one has to do with the endogenous cannabinoids that we and all mammalians produce, anandamide and 2-AG, two compounds that we discovered in the 90s. And since then, there has been a lot of interest, at least amongst many scientists, of compounds that we produce, mammalians produce. There are about 100 of them. They are like anandamide. Anandamide is a relatively simple compound, a fatty acid bound to an amino acid or an amino acid derivative. And there are about 100 compounds. And many of these, at least those that have been investigated, are of major importance. And I don't believe that the body makes compounds because it has nothing else to do. So we have about 100 compounds, and these are being investigated. And I'll spend a part of my talk on these compounds. So let's start with the phytocannabinoid research. I was surprised to find out in the early 60s when I started work that contrary to morphine, which had been isolated 100 years previously from opium, and contrary to cocaine, the cannabinoid chemistry was not well known, although some excellent chemists had worked on that. And this is because it turned out that there is a huge number of cannabinoids in the plant, and the techniques were probably not available in the 30s and 40s to identify these compounds. And so we went back. I got, without any major administrative problems, I got 5 kilograms of hashish, Lebanese hashish from the police, and we started looking at the cannabinoids there. Incidentally, the hashish I got was smuggled hashish, which Lebanese and Israelis collaborate to get through the frontier, but only part of it is being caught, and that came to my lab ultimately. And we started isolating the materials there, and we were able to isolate about a dozen compounds, most of them unknown, and all of them, for those of you who remember your high school chemistry, the right-hand side of the molecule is more or less the same. Now we know there are some differences, but at that time, we found out that all the compounds we identified had the right-hand side, the aromatic part of the molecule, if you wish, the same, and the differences were only in the left-hand side of the molecule, which is a terpene, and these modifications led to a huge number of compounds, and they are identified, at least some of them identified here, cannabigerol, cannabinol, THC, etc., etc. But the two most important compounds were CBD, cannabidiol, and THC, which we elucidated the structure of cannabidiol, which had been isolated way back in the 30s, but the structure was unknown. And we published that in 1963. That was our first paper on cannabis. And THC, we were able, I believe, for the first time to isolate in a pure form and to elucidate its structure and its absolute configuration, the way it stays in space. And these two compounds are the two compounds that have been investigated most thoroughly. And there is still quite a lot of work on some of the additional compounds. We don't know enough about cannabigerol. We don't know enough about cannabichromine. We certainly know almost nothing about cannabis cyclol and so on and so forth. So it is worth doing. But all the other compounds, to the best of my knowledge, except THC, do not bind to the cannabinoid receptors, and I'll have a few words to say about it. But with cannabis, everything is complicated. We turned out that THC, CBD, all the other compounds are not actually natural products. They're secondary products. They're made in the plant from their acids. The plant produces CBD acid, THC acid, cannabigerol acid. These compounds are not stable, and they convert on the plant itself or later, they convert into their neutral compounds. So cannabidiol acid converts into the cannabidiol, and THC acid converts into THC. Now, there isn't much work on the acids because they're not stable. And scientists don't like to work with unstable compounds because one doesn't know whether the results are due to the starting material, to the acid itself, in this case, or to those compounds that are derivatives of cannabidiol. So there is not enough work on the cannabinoid acids. So we thought over the last few years that it's worthwhile to stabilize the acids and then maybe look at their activities. And indeed, as I mentioned, over the last few years, we have been working particularly on cannabidiolic acid, which we stabilized by converting it into the methyl ester. And this compound is stable, and this compound apparently, to a certain extent, parallels the activity of CBD. In some cases, it's more active, and we have already published several papers on the activity of this particular compound, and we are going to hear a few lectures on this compound as work in this field is being supported by one of the two groups that's organizing this meeting, EPM. And I believe that after we know more about CBD acid methyl ester, it will, I hope, hopefully become a drug. What do we know about the cannabidiolic acid methyl ester? Well, we found and published that it suppresses nausea. As you know, cancer chemotherapy does cause nausea, and sometimes most unpleasant. It suppresses nausea, and it suppresses anxiety in rats. We don't know whether it suppresses the anxiety in humans because it has never been given to a human, never administered to a human, and we are neither rats nor mice, so we don't know. But this particular compound was found that it enhances serotonin activation. As all of you know, serotonin, dopamine, these are the two major neurotransmitters, and they're of utmost importance in a large number of diseases. So this is probably one of the mechanisms that the acids and the neutral compounds act. And in any case, this compound, 580, the methyl ester, was found to lower anxiety. And quite recently, we also found that it lowers, again, in animals, lowers depression. Again, we can never say 100% sure whether depression in animals parallels depression in humans. I believe that this is so, so I'm looking forward to see whether the methyl ester will suppress anxiety and depression in humans, and I hope that the next meeting here, the next few meetings here, will hear a lot about these compounds. So before I go into some detail, I want to say a few words about an effect which we observed quite a few years ago. We called it the entourage effect. And we found that compounds that are present in cannabis and do not have any significant cannabinoid activity by themselves affect the activity of the cannabinoids of the plant. They affect the activity of CBD, they affect the activity of THC. We published that, and there are a lot of papers that refer to the entourage, saying patients prefer to use a cannabis extract rather than pure THC or pure CBD because of the entourage effect. This may or may not be fully correct, but there is surprisingly very little work on the entourage effect by itself. We have a huge number of terpenes, we have flavonoids, we have other compounds in cannabis, and we still do not know how these compounds, or which are these compounds in the long list of terpenoids and flavonoids that cause the entourage effect. And I hope, again, that more work will be done on the entourage effect because it is an important observation, I believe, and it does not really answer all the questions we have, but it is of major biological importance. So let me say a few words about the effects of THC. They have been published, they're well known, so I will not spend much time on THC. Just a few of the major effects. We know that it affects pain, nausea and vomiting, sleep. A lot of patients report that sleep, allowing them to go to sleep, is the major effect that they can see, and it lowers appetite. And again, as we all know, at low doses it may cause these effects, but if we go to higher doses, especially people that have not used cannabis, THC previously, if they use initially high doses, many of the mostly young people, end up in the psychiatry department with an attack. And indeed, I have found out that in the hospital I'm associated with, the medical faculty of the Hebrew University, the psychiatrists are strongly opposed to cannabis. And the reason is that they see almost every day 1 or 2 young people being brought to the hospital with an attack because they started using initially large amounts of cannabis, large amounts of THC, which of course can cause a psychiatric attack. Most recently, we have seen that THC, again, at low doses can lower depression, and of course many patients say so. But again, we have a biphasic effect here. At low doses, we definitely see antidepressant effects, but at higher doses, we don't. Higher doses, we know the effects. The other cannabinoids do the same, but it seems that they do that at much higher doses. And some years ago, we looked at the possibility that THC is effective in post-trauma. Of course, a lot of post-trauma patients use cannabis, and we wanted to know whether this is true or not. So we did a small clinical trial, and we found that, yes, most definitely, it affects most of the symptoms of post-trauma in patients. And we had patients that some of them were soldiers previously and had seen people around them get hurt. There were a few women that had been raped. There were a few people that had been in traffic accidents. So we had patients of all types. And yes, most definitely it helped with the post-trauma, and I hope that there will be a much larger trial, because a lot of patients, as you know, a lot of post-trauma patients use cannabis, and we really have to know more. But we don't have enough clinical trials. And again, I want to stress, we badly need clinical trials. This is the one thing that physicians insist on, rightly so. They can base their work on clinical trials, and we don't have clinical trials for most of the diseases that are well known to be affected by cannabinoids. I have not seen yet a major clinical trial in cancer, any cancer. And we know that some cancer affected more, some cancer affected less, but we don't have the data. So I strongly ask the clinicians here, please try to go ahead with modern clinical trials in most of the diseases that we're speaking about, certainly in cancer. Well, what about CBD? Well, of course, CBD is well known. I was surprised to see that people think that CBD will be used to the extent of $25 billion a year within the next few years in the US alone. It may be an exaggeration after all, but I'll say a few words about the effects of CBD. We and many others had seen in animals that CBD, pure CBD, affects epilepsy. So we did a clinical trial, as you can see, we published it in 1980, almost 40 years ago. And this was done in collaboration with my colleagues in Sao Paulo in Brazil. And we supplied them with almost 500 grams of cannabis, of CBD, we had isolated from cannabis. And they gave relatively high doses, 200 milligrams a day, to epileptic patients, that nothing else was helping them. And they found that, yes, indeed, it is most definitely a very helpful drug. It was done on a small number of patients, but even so, that should have meant at that time that, yes, here we have a drug that can help with epilepsy. Well, what happened was nothing. Nobody was interested. So for nearly 30 years, nobody used cannabidiol in epilepsy. But in the US, parents saw that cannabis, the plant, cannabis that contains the high doses of CBD, is indeed helpful, especially in children that had epileptic attacks, sometimes 20 to 30 epileptic attacks a day. This was the only thing that helped them. So they started going to one of the states that cannabis was legal, and there was a lot of public pressure, and the U.S. Food and Drug Administration ultimately approved a clinical trial. Until then, I don't believe that there was any major clinical trial with cannabinoids in the U.S. Well, as we know, the trial was a relatively large trial, was very successful, and today CBD, in the name of Epidiolex, is being used as an anti-epileptic drug. Did we have to wait 30 years? No. We could have helped children, in particular children, over these 30 years. Having 20 to 30 epileptic attacks a day really means that the child of this type cannot develop. We ruined the child. We could have helped maybe thousands of children. We didn't. And I'm under the impression that this is what's happening with quite a few of the other diseases. In one field, we have pretty good information, and this is the graft versus host disease. We and others had found that cannabidiol works in autoimmune diseases, namely diseases in which the body attacks itself. The reason why it does that, quite a few, but this is the ultimate fact. The body attacks itself in many autoimmune diseases. I've been told that there are about 80 of these, about 10 are pretty well-known and serious. And a colleague of mine, Dr. Yeshurun, thought that maybe he should try cannabidiol in something that is closely related to autoimmune disease. Namely, in certain cancer cases, there is a bone marrow transplant, and the body doesn't like that, and the transplant doesn't like the body, and they attack each other, and the patient is sick. It's sometimes very sick. If this is a kind of an autoimmune disease, then maybe cannabidiol will be helpful. And indeed, he tried pretty high doses, about 300 milligrams a day of cannabidiol. He gave it to these patients, and indeed, he helped them. He helped most of the patients. Here are the results that he had. He had about 100 controls, and 46% of these had a mild attack, a relatively mild attack, and 10% have had a very serious attack. When those that got CBD, only 12% had unpleasant symptoms, and out of those that the serious patients, which previously were 10%, went down to 5%. So now, cannabidiol in GVHD is in the, I think, third phase investigation, and I sincerely hope that it will become a drug within the next few years. And again, I hope that it will be possible to help quite a lot of patients. The third example I want to give is schizophrenia. We had done a small trial of CBD, high doses in schizophrenia in Brazil many years ago. And it was quite successful, but it was not taken up, and it was just an interesting paper in the literature, but not more so. But in Germany, Dr. Levicke did a major clinical trial, and he found that indeed CBD was was a very good anti-schizophrenia drug. He compared it to another drug that was being widely used, and both drugs were active. But the activity of the drug that's presently being used had a lot of side effects. CBD, as we know, does not cause side effects. So here we have a drug that apparently helps a lot with schizophrenia. And he saw, and this seems to be the mechanism, CBD enhances anandamide levels in the body. So maybe this is the way it works. I'm not sure at the moment. We don't have additional information, but this is what's happening. And yet, although the paper was published 7 years ago, it is still not being used in schizophrenia. Again, are we missing something? We don't have enough data. And obviously, psychiatrists will not be using CBD in schizophrenia without additional data. We may be losing something important. Again, please, those of you, if there are any psychiatrists here, try to repeat and try to enhance the work that was published by Levicke and then by others later on, and we need the data. Now, Levicke found that when cannabidiol was compared to the drug that was used quite a bit, amisulpride, it caused less weight gain, which amisulpride causes. There were no changes in hormones, and there were few extrapyramidal syndromes, and that means that there were less muscle spasms, there was less slowness of movement, there was less tremor, and there were less irregular jerky movement. So what I'm trying to say, here we have a drug which possibly, quite possibly, acts well on schizophrenia, and we are speaking about 1% of the population which has schizophrenia, and a large part of this population does not work, cannot study. It is, to a certain extent, lost. We can help, I hope. And I want to mention, speaking of the cannabinoids and the plant cannabinoids, again, another autoimmune disease, diabetes type 1. It is an autoimmune disease, and we have a good model in animals, so we try to see whether cannabidiol helps with that. And in the diabetes type 1, the body destroys the cells that produce insulin. And indeed, those mice that were not treated with CBD after a certain period of time lost most of the cells that produce insulin, while those that were CBD treated, 77% of the cells remained intact. We badly need new diabetes type 1 drugs, but it has never been tested in the clinic. Well, schizophrenia, at least we have 1 or 2 clinical trials. With diabetes type 1, we don't have a single one. Why? I have no idea. Now, if we speak of the mechanisms of CBD, here we have a major problem. It seems to bind to a lot of receptors. It has all kinds of mechanisms. At the moment, I prefer this mechanism, which was published recently, that CBD causes an increase in certain cells that are called MDSC cells, and these cells are known to suppress another type of cells that cause inflammation. They're called CD4 T cells. Whether this is correct or not, whether this is the only mechanism, I don't know, but it seems to be to make sense. Cannabidiol seems to cause lowering of the activity of these CD4 T cells. We'll have to wait over the next few years to find out whether this is true or in all cases, only in some cases. Now, many of the drugs that we have on the market are not natural products as they appear. They are derivatives. You cannot buy penicillin in the drugstore. The penicillin is not a drug. Derivatives of penicillin are drugs. The same is true for cortisone. You cannot buy the hormone cortisone in a drugstore. You can buy prednisone or derivatives of cortisone. So I think that this may happen to a certain extent with cannabidiol and related compounds. And indeed, if we take cannabidiol and make a small change, put a fluorine atom on the cannabidiol, it is more active than cannabidiol itself. And as I mentioned before, the methyl esters of the acids do the same. And chances are that we shall have, within the next 10, 15 years, on the market, both cannabis, the plant materials, and derivatives like the fluorine compound, like the methyl esters and so on, which is not a bad situation because patients will be able to choose whatever they believe is best for them. Now, up till now I talked about the plant phase of the cannabis. It started to change when the mechanism was, there was research on the mechanism. In the 80s, namely almost 20 years after the cannabinoids were identified, an American group, Alan Howlett, discovered that there is a receptor, and this receptor, which is a specific one, is found in those areas of the brain that are quite typical for cannabinoid, for THC effect. Those areas of the brain that have to do with movement control, with coordination, learning and memory, cognitive function, and so on. So here we had an opening to the mechanism by having a receptor. And today we know that we have two receptors. But receptors don't exist in the body because there is a plant out there. Otherwise, we'll have millions of receptors. Receptors exist because we produce compounds that bind, that stimulate or inhibit these receptors. And so we went ahead looking for the compounds, the natural compounds that do that. And how does the one do something like that? Well, for those of you that are interested in the chemical part, we take THC, for example, put on a tritium atom and on it, and the compound is still very active. And then we bind this tritium labeled THC to the receptor, and then start to look at the areas of the brain that also want to bind to the receptor, and doing so they kick out the labeled material, and we can measure the labeled material that has been kicked out from its binding to the receptor. So by this simple way of doing it, it takes only 3-4 years and a few PhD students and quite a lot of others, we were able to identify two compounds. One of them we call Anandamide, and the other one 2-AG. And these compounds are essentially the cannabinoids of our body. They are chemically completely different than THC, but their activity is essentially the same. THC mimics the activity of the compounds that we make, the compounds Anandamide and 2-AG. And there are, of course, some differences. These compounds are made in the body when and where needed. Particularly, anandamide is not present in large amounts. In the body, it's present in very small amounts. But it is synthesized by the body when and where needed. It acts, and then the body breaks it down essentially immediately. So we don't see the effects that are so well known with THC, as THC stays in the body for quite a long time. So as here we have two neurotransmitters, novel neurotransmitters. I will, of course, not go into the biochemistry, but contrary to most of the other neurotransmitters, they, as I said, are not present in the body in large amounts. They are present in the nerve. The nerve is not just a wire going from my hand to my brain. There are breaks in the middle so the body can regulate it, and these are called synapses. And anandamide and 2-AG are formed in the postsynapse, the part of the brain that's after a break, and they go out from the postsynapse to the presynapse. They activate the receptor, and then they affect the formation or the release of other neurotransmitters. So what I'm saying in short is that these compounds, and THC also, act not only by themselves, but they also act by regulating other neurotransmitters. And this is one of the reasons why we see so many effects by both THC and the endogenous cannabinoids. So the endogenous cannabinoids, anandamide and 2-AG, are very, very important and interesting compounds. But again, because of all kinds of legal problems, they have never been administered to a human. That's quite frankly outrageous. When insulin was discovered almost 100 years ago, it was given to humans within a couple of months and became a drug within a year or so, an excellent drug. Insulin, derivatives of insulin, are being used 100 years after discovery. Anandamide and 2-AG, which were discovered in the mid-90s by our group, have never been given to a human. Although they are not toxic, they do not cause any major side effects, but because of the legal situation, nobody has given them to a human. I have no solution to that. What is the endocannabinoid system? Now, I mentioned that we have two receptors, CB1 and CB2. Activation of the CB1 causes the well-known cannabis effects. The activation of CB2 leads to essentially to protective actions. It is kind of a receptor that parallels the immune system. It helps in a large number of disease states, and I'll say a few words later. Then the endocannabinoid system contains anandamide and 2-AG. The enzymes that synthesize these compounds, the enzymes that break down these compounds, and quite a few companies are working on these enzymes because they can enhance the presence of anandamide, let's say, by blocking the enzymes that destroy these compounds, that metabolize these compounds. And so I expect that a few new drugs will be available on the market within the next 5-10 years, because these compounds can affect the levels of anandamide and the levels of 2-AG by playing around with the enzymes that synthesize them or the enzymes that break them down. Not directly, not giving anandamide directly, not giving even a derivative anandamide directly, but by playing around with their being built up or destroyed. Now, a lot of scientists have looked into the activity of anandamide and 2-AG. There are about 6,000 publications that have to do with anandamide and 2-AG. And today we have quite a big list of compounds that physiological state that are affected by anandamide and 2-AG. And here is an alphabetical list, and starting with anxiety and blood pressure and immune system, etc., etc. If I had a full list, it will take another 2-3 slides. So here we have a system of major importance, and I have no doubt that within the next few years we shall see many more papers. In addition to the about 6,000 papers that have been published, we'll see many more papers, and this will open to us or clear to us many of the physiological problems that we know exist, but we have not been able to show, to understand how they happen. Now, I will give just one example of work that I did with one of my colleagues, Professor Shohami. she's a specialist in brain trauma. And we wanted to know what happens with the endocannabinoid system in brain trauma. So she caused brain trauma mice, and we found that the 2-AG in the brain goes up tremendously. The 2-AG is the red thing that you see in the middle. If you see and compare it to the controls, you'll see that there is enhancement, very, very potent enhancement of 2-AG levels in the brain after brain trauma. Is that because we have caused brain damage and the brain doesn't know what it does, or is it a reaction of the brain, kind of a protective reaction, if you wish? So we synthesized a large amount of 2-AG and gave them to mice that had undergone brain trauma and compared them to mice that had not undergone brain trauma. And we found that the 2-AG lowers by 50% the infarct volume in the brain. The infarct was not a very strong one. It caused a certain amount of damage, but it could be compared. So we saw that most definitely the brain reacts to the damage by producing 2-AG and lowering the damage. Again, we see quite a lot of people with brain damage. Maybe 2-AG is a good protective compound, protective drug, if you wish, but again, nothing has been done in this area. Now, I mentioned to Dr. Shohami, whom I collaborate. I still collaborate in many areas. In the field of cannabis, whether it's the plant cannabinoids or the endogenous cannabinoids, there should be a lot of joint work, a lot of collaboration. There is no way we can understand what's going on by looking just at the chemistry, or looking just at the pharmacology, or looking just at the physiology. It has to be in collaboration, and our group has collaborated with a huge number of colleagues all over the world, and I mention that in more detail around the end of my lecture. Now, here is what I said, that 2-AG, in addition to blocking the brain trauma, or at least relieving some of the damage that was caused, was found to be decreased in major depression. So is it possible that, at least in some patients, major depression is caused by the fact that the body has levels that are too low, levels of 2-AG? Now, we have seen something of that sort between anandamide and anxiety, and this was work that was published by Hill some time ago. Again, here we have an opening that most definitely needs further development. And as I said, there are two receptors. The CB1 receptor causes, in addition to many other things, the well-known effects of cannabis, changes in mood and changes in activity and so on. The CB2 doesn't do that. CBT is exclusively a neuroprotective mechanism. At least 90% of its actions are protective. And one can see that because there are papers showing that CBT receptors work in myocardial infarction, in atherosclerosis, in stroke, in fibrosis, etc., etc. Again, there are a few companies working on synthetic CB2 agonists, and I believe that we shall see synthetic CB2 agonists as drugs against at least some of these diseases. And it is possible to do that because we have, and other groups have done the same. It is possible to synthesize compounds that act only on the CB2, on the protective mechanism. They don't act on the CB1, so it doesn't matter whether somebody gets very high doses. One does not see the cannabinoid effects. One sees only the effects on the CB2. And it is possible. Anandamide, 2-AG, THC act both on CB1 and CB2. It's possible, again, as I said before, to synthesize compounds, and here to examples of compounds that act only in the CB2, and chances are that we shall see the compounds of this type in the market. Now I want to come to the last part of my lecture, and it has to do with the activity of anandamide-like compounds. As I said, anandamide is a simple compound, relatively simple compound. It is formed from a fatty acid which is bound to an amino acid, and our body has about 20 fatty acids there, and has about 20 amino acids, and so the body, using already the synthetic mechanism it knows, our body is lazy. If it can use something that it knows how to use, it will use it as much as possible. So in this case, the body has learned how to make anandamide, and by the same synthetic mechanism, it can make many other compounds, fatty acids bound to amino acids, and it has made about 100 of them. So I'll speak about the 3 or 4 that have been investigated. Now, let's start again from brain injury. Brain injury acts, I've been told, in 2 directions, causing the effects on compounds present in the body called glutamate and cytokines and so on, and they can bring to cell death. And this is what we see in most cases of brain injury, but there is also something else. In brain injury, one sees vasoconstrictions. The blood vessels constrict, and this is not healthy. And so the body tries to prevent or to lower vasoconstriction. So we compared the vasoconstriction whether the THC, CBD, or anandamide, lower vasoconstriction, and we saw that the brain extract does that better than these compounds. So the answer was, do we have compounds that the brain makes and that they block or try to block vasoconstriction? They are vasodilators. So I had a PhD student who worked very hard on that, and after a few years was able to identify a compound which is very close chemically to anandamide. You can see the anandamide on the top, on the left-hand side, and we isolated a compound which is essentially the same, but it has another carboxyl group there. It does not bind to the receptor, but it is a vasodilator, a pretty potent vasodilator, and this is what our body does when there is a vasoconstriction. It synthesizes this compound which is closely related to anandamide. So we said to ourselves, well, maybe there are additional anandamide-like compounds in the body. So this is the 3rd phase, the phase which quite a few people in the field work. The 3 phases I mentioned are the plant chemistry and the plant cannabinoids, the compounds that we make, anandamide and 2-AG, and now I'm speaking of anandamide-like compounds, and this was the first case when we discovered that rachidonylserine has something to do with vasoconstriction. We were doing some research in Professor Babb's lab on bone remodeling. It had nothing to do, at least we thought it has nothing to do with bones, and we shall hear quite a bit on that by a colleague of ours who will be talking this afternoon. And we found that the body makes again a compound which is again an anandamide-like compound. There is a fatty acid, in this case oleic acid, found in olive oil, and it is bound to an amino acid called serine. Now, all this came from a work that was published many years ago showing that osteoporosis is less prevalent in the Middle East or in the countries around the Mediterranean than in Norway. And that's what they suggested, maybe because women in the Mediterranean region consume olive oil, and indeed after the age of 55 and so on, with all the hormonal changes, the body does something which prevents the osteoporosis, which otherwise will be seen in essentially all women at that age. And so we tried olive oil and we tried oleic acid. They didn't work. But then we tried derivatives of oleic acid, and we then looked for these compounds in the body. And we found again, as I said, a fatty acid bound to an amino acid. It is a very potent compound that works against bone destruction. There is company that thinks that it can do something with it and developing it. So here we have a compound which may prove to be of major interest. Indeed, if we take animals and cause osteoporosis, and I wouldn't go into the details, you can see the red, the bone is being destroyed, the green is before the destruction of the bone, the red is after the bone has started to be destructed because of the hormonal changes. If we give these animals the compound, synthetic in this case, of course, the compound that our body makes, the bone starts growing up. Now, this may be of importance because most of the drugs that are available today for that kind of disease stop the destruction, but they do not, in most cases, help with the new formation of bone. And here we have a compound that maybe also in humans, they have never tested in humans, not only stops the disease, but also causes the bone to be built again. And I want to end with something that we're doing at the moment. And it has to do with addiction, not cannabinoid addiction, but addiction in general. Our body sees addiction as a disease. For many years, it was thought that it is something only psychological, nothing to be seen chemically or biochemically or pharmacologically. Well, today this is not accepted anymore. It is addiction is considered to be a disease. If it is a disease, the chances are, we thought, that our body tries to fight against the disease. This is what our body does with most diseases. So how does our body fight addiction? And turned out that we do not know. We do not know anything. Most of the research that has been done on addictions is on the addicted people. Nobody has looked into those that use compounds that cause addiction and are not addicted. Why aren't these people addicted? And why not addicted? Well, again, this was done in collaboration. As I said before, collaboration is essential to work in the cannabinoid field. And in this case, were 4 groups, in addition to mine, a group in Virginia, the US, a group in Canada, and a group in Italy, and we meet at meetings, we drink a lot of coffee together, we discuss things, we don't throw the chairs one to the other, but we discuss things for many hours. And we decided to look for compounds that maybe our body produces. And we found a paper that a person who was strongly addicted to nicotine and a certain part of his brain had been damaged in a car accident was not addicted anymore. So we thought maybe this part of the brain produces a compound that blocks nicotine addiction. Well, nobody volunteered to give his brain, so we had to do it with mice again. So we had mice that were addicted. We had mice that had brain damage in that particular part of the brain, and we looked for the compound, and indeed we found the compound, and to our great surprise maybe, we found that the compound belongs to the Anandamide-like compounds. It is a relatively simple compound, oleic acid bound, which is a fatty acid, bound to glycine. And this particular compound blocks the formation of nicotine addiction. There are quite a few ways of doing that. One can, just in a few words, one can have mice sitting between food that contains nicotine and food does not contain nicotine, and this is called conditioned place preference. Then the mice at the beginning go 50% here and 50% there, but then they slowly get addicted to the nicotine, and all of them go to the part of the where the nicotine is. So we can block this and a few other models by administering allele glycine to these animals. Then they don't get addicted. Does this compound work in other addictions? To the best of our knowledge, the answer is no. We tried to see whether this compound, OG, allele glycine, can block the same thing with opiates. It does not. But we found something else. We found that these particular compounds at reasonably high levels can block the withdrawal symptoms both of nicotine addiction and of opiate addiction. And our colleagues in Canada have now done quite a lot of work, outstanding work, I believe, showing that this simple compound can block the withdrawal symptoms not only of nicotine but of morphine. Sometimes, very often, one of the treatments of opiate-addicted people that are brought to the hospital are given a compound that will, naloxone, that will cause withdrawal, but it can help the patient, but the withdrawal can be really, really very bad. Well, we can block the addiction, at least in mice, by giving them the allele glycine. Now we're trying to find out whether we have additional compounds made by the brain in mice, brain of rats, that work against the opiate addiction. Because we know that not all users, opiate users, get addicted. 80%, 90% of them do. What about the others? So I believe that here we have quite an opening to some new, ideas maybe, and hopefully these ideas will bring new drugs against the various addictions. Well, let me summarize what I've been saying over the last almost hour. The endocannabinoids are involved in a large number of physiological processes, and THC mimics their actions. I'm surprised that the endocannabinoids and the endocannabinoid system were not discovered when dopamine and serotonin were discovered. These are important neurotransmitters. Why? I don't know. Because they are not more complicated than the dopamine, the serotonin system. But this is what we have. So over the next decade, few decades, we shall learn quite a lot more about the endocannabinoid system in this respect. On the other hand, CBD has turned out to be an extremely interesting compound. And again, we should know more about CBD after clinical trials are done. At the moment, we see a lot of noise around CBD, but in most cases, we have no clinical trials, and many of the things we hear about CBD are just, out of the blue, if you wish. Now, the third thing that I want to mention, that the endocannabinoid-like compounds, and this was the third part of my lecture, may certainly bring new drugs to a wide variety of diseases, and I hope that this will happen. And the same is true for the CB2 specific compounds that act on the CB2 receptors alone, because we see no side effects, except in very, very unusual cases. We see no side effects from drugs that act on CB2 alone. And I want to thank my collaborator in Israel, many of them in my lab. Some of them are professors in different departments. They may be in the School of Pharmacy, some of them are in the School of Medicine. And I want to thank in particular my collaborators abroad. I said it is impossible to do work in this field and actually in many, many other fields by just working in one part of the area, only in chemistry, only in pharmacology, or something of that sort. It has to be done in collaboration, because I see no difference, as a matter of fact, there's no scientific difference between chemistry and pharmacology. We separate them because we are possibly unable to know everything about two fields. We barely know a little bit about one of the fields. And I want to thank them, and I want to thank you for being at my lecture.